will begin our class now. So today also we are continuing with the same unit, plasma membrane. Okay, so today we are going to discuss exoosmosis and endoosmosis. On the other day, just I clarified you what is exoosmosis and what is endoosmosis, and today we'll see with the help of one practical experiment. Okay, so that activity is to prove exoosmosis and endoosmosis. Aim of the activity, just write down first. Write down the aim of the activity to prove exo and endoosmosis. To prove exo and endoosmosis. This is the aim of the activity. Then materials required. Materials required. Materials required to write down 3 beakers, 2x rice, then dilute hydrochloric acid, then dilute hydrochloric acid, then tap water. Tap water. You take tap water. Okay, so these are the materials required for the experiment. Aim of the activity is to prove exo and endoosmosis. Materials required to do this activity is 3 beakers, 2x dilute hydrochloric acid, or you can also take these acid. Toilet cleaners. Any toilet cleaner you can take. Okay, and tap water. Then next what you have to do is procedure. So take 3 beakers. In 1 beaker you take tap water. In the remaining 2 beakers you take dilute hydrochloric acid and keep 2x for 4 to 5 hours. Keep 2x for 4 to 5 hours. You take this 2 beakers. In that you take dilute hydrochloric acid or any toilet cleaner. Okay, you take any toilet cleaner and keep the 2x raw x for 4 to 5 hours. Then take the measuring scale, or with the help of paper also you can prepare. Okay, then keep it for 4 to 5 hours. After that, remove this 2x and keep it on the paper and measure the 2x. Measure the 2x with the help of scale, or you can take paper scale also by preparing from your notebook paper. Okay, then after this, what you have to do is whatever egg you have taken in 4 to 5 hours, take that egg and dilute hydrochloric acid. And next, what you have to do is keep 1 egg in dilute hydrochloric acid, the other egg in tap water. The other egg in tap water. Okay, this eggs which you have kept in already in dilute hydrochloric acid, that egg, 1 you take in tap water, other you take in concentrated salt solution. So what you are doing first, you are keeping the 2 eggs in dilute hydrochloric acid or any toilet cleaner for 4 to 5 hours. Then measure the circumference of 2x. Measure the circumference of 2x. 2x are similar in size. Then what you have to do is take this 2 beaker. In 1 beaker you take tap water. In another beaker you take the salt solution. In 1 beaker you take tap water. In another beaker you take salt solution. So again the eggs which you have kept already in dilute hydrochloric acid or toilet cleaner, that 2 eggs you keep in tap water and salt solution for 2 to 4 hours. For 2 to 4 hours. Okay, so this is our procedure. What you have to do in procedure, take 3 beakers. In 1 beaker you take dilute tap water, another beaker you take dilute hydrochloric acid in 2 beakers. In 2 beakers you take dilute hydrochloric acid and keep the rice. Measure the circumference is same. Then that 2x you take and take the 2 beakers. In 1 beaker you take tap water, in another beaker you take salt solution. Keep these 2x which you have already kept in hydrochloric acid in this 2 rice. Okay, we kept this in hydrochloric acid. This you keep in tap water and salt solution for 2 to 4 hours. Okay, then keep it for 2 to 4 hours. Then after that, start your observation. So what did you observe already? We measured the circumference of the egg. It was similar when we kept it in dilute hydrochloric acid for 4 to 5 hours. Then when we are keeping this in different solutions, that is in tap water and salt solution, and when we are removing these two eggs and when we are seeing one egg will undergo the shrinkage, this the egg will become small, the other will undergo the enlargement. This will become big in size. Okay, one egg which is kept in tap water and salt solution, here we are seeing observation, one will undergo the shrinkage, will become small in size, the other will undergo the swelling, the other egg will undergo raw eggs, not boiled eggs, the other will undergo the enlargement, which will become big in size. This is our observation. So, what do we conclude by this? We are concluding that why it is undergoing swelling is, it is taking the water inside. It is taking the water inside. Such a process of taking water inside the egg is called as endoosmosis. Endoosmosis. The egg which is kept in salt solution, it is taking the water inside and undergoing the swelling. So such a process is called as endoosmosis. And the egg which is kept in tap water, it will lose the water from it and it will undergo the shrinkage. Such a process is called as exoosmosis. Okay, exoosmosis. By this activity, we are doing the endoosmosis and exoosmosis. Leaving the water molecules from the substance is exoosmosis, and taking the water inside, it is called as endoosmosis. Okay, so by this activity, we are doing exo and endoosmosis. Okay, so next activity is next, with the same egg, the egg which you kept in dilute hydrochloric acid, you take the same egg. You take the same egg to know how to prepare the semi-permeable membrane. Preparing semi-permeable membrane. Preparing semi-permeable membrane. How to prepare the semi-permeable membrane. So before going to this activity, I want to know how many of you have eaten the boiled egg. So while you are eating the boiled egg, while you are removing the shell, you are getting a thin membrane over it. Between the shell and the egg, you get a thin membrane like onion peel. So that is nothing but it is plasma membrane. So with the help of raw egg also we can prepare the semi-permeable membrane. How we prepare the semi-permeable membrane means, in this previous activity, the raw egg which is kept in hydrochloric acid, keep that egg for 4 to 5 hours in hydrochloric acid and slowly you puncture it. The egg will break out. Shell of the egg will break out. So the shell of the egg is made up of calcium carbonate. Shell of the egg is made up of calcium carbonate. Okay, so the shell of the egg is made up of calcium carbonate. So when you are keeping this egg in hydrochloric acid for 4 to 5 hours, and when you are just piercing it, the shell will easily break out. Why? Because you are keeping it in hydrochloric acid or any toilet cleaner. So the shell of the egg will easily break out. Then, why? Because the egg shell is made up of calcium carbonate. Then you will get a thin membrane over it. You will get a thin membrane over it. That membrane is nothing but permeable membrane, semi-permeable membrane, or plasma membrane, or cell membrane of the egg. Egg is one cell, so it is surrounded by the cell membrane. That is, when you eat boiled egg, you are observing. You know, even if you did not observe, now you observe how this membrane is there. That is nothing but it is called as plasma membrane. So you have to take 2 beakers again. In that 2 beakers, you take hydrochloric acid, take 2 eggs, and keeping these 2 eggs in hydrochloric acid for 4 to 5 hours. Same previously what we did. That eggs also we can take and just to remove the shell. Easily the shell will break out. Easily the shell will break out. Then with the help of pencil, just puncture the plasma membrane. Small hole you make in the egg membrane. Okay, you make a small hole with the help of a pencil tip, or you can take the pin also and puncture it. Drain the entire matter, yolk, white of it, albumin, all you remove from it. Remove the entire content of the egg inside. You remove yolk and albumin which is there. You remove it. You drain it. Okay, you drain these 2 eggs completely, then you will get a thin membrane, that is nothing but it is called as plasma membrane. Then to observe the osmosis process again, what you are doing is taking these two egg membranes. In one egg membrane, you are taking 10 ml of saturated sugar solution. 10 ml of saturated sugar solution in one egg membrane. And in another egg membrane, you are taking 10 ml of water. In one egg membrane, you are taking saturated sugar solution, and in other membrane of the egg which you prepared, you are taking water. Then tie the mouth where you have made the hole there. You have to tie the mouth with the help of thread. Then your doubt, you may get a doubt here that how can we fill this in the egg membrane, because egg membrane is very transparent and very delicate. How can we fill it? With the help of syringe. Syringe, you know, we take injection. That syringe you take and you fill 10 ml of saturated sugar solution in one syringe and 10 ml of water in other syringe. The two syringe, you just fill it in the egg membrane. Okay, then tie the mouth of the two egg membranes with the help of thread. Okay, then it will feel like a balloon. Okay, like when you are blowing air in the balloon, how they appear, in the same way they will appear. Then what you have to do now is you take beaker, two beakers. You take two beakers. In one beaker, you take tap water. In one beaker, you take tap water. In another beaker, you take saturated sugar solution. In one beaker, you take tap water. In another beaker, you take saturated sugar solution. Now, what you have to do here? Listen carefully now. So, where the egg membrane in which you fill sugar solution, that egg membrane will keep in tap water, and the egg membrane in which you fill tap water will keep that egg membrane in saturated sugar solution. 100 ml of saturated sugar solution you are taking here, 100 ml of tap water you are taking here. Okay, 100 ml of water you are taking, 100 ml of sugar solution you are taking in two different beakers. In the same way, in two egg membranes you have taken here, in one egg membrane you have taken, you are tying here, in one egg membrane you are taking tap water, and in another egg membrane you are taking sugar solution. Okay, in one egg membrane you are taking tap water, in another egg membrane you are taking sugar solution. So the egg membrane in which tap water is there, you just keep it in this, and this egg membrane in which sugar solution is there, you keep it tap water. Then keep it without disturbing for 2 to 4 hours. Without disturbing, you keep it for 2 to 4 hours. Then after that, observe the 2. So one egg membrane where the saturated sugar solution is there, it will undergo the shrinkage, and where the tap water is there, it will undergo the bulging. Why this is happening? Movement of molecules movement of solvent is taking place. Water is nothing but solvent. Movement of solvent takes place into the solute, that is sugar solution. From the beaker, it is going into the egg membrane. As a result, the egg membrane will bulge. And here, the egg membrane is undergoing the shrinkage because it is losing the water into the sugar solution, and it is undergoing the shrinkage. It is becoming small in size. So, by this experiment also, we are understanding how plasma membrane is playing a major role, acting as a semi-permeable membrane, selective permeable membrane, okay, impermeable membrane. In all the ways, how the cell membrane is protecting the cell. By this activity also, we are doing the same. Okay, so that is about the plasma membrane and its activities. Now, we'll discuss the next topic, that is reverse osmosis. Next, we'll discuss reverse osmosis. What is reverse osmosis? Okay, next we'll discuss reverse osmosis. Okay, Before going to reverse osmosis, just tell me what are the functions after studying the complete activities, what are the functions of osmosis in our daily life as well as in our physiological process. So if you take in our physiological process in plants and animals, the water from the root system is entering into the entire plant body by osmosis process only. Water move between cell and cell. Water and minerals move between cell and cell in the plants by osmosis process only. Not only that, it also helps for closing and opening the stomata. It also helps it closing and opening the stomata. Okay, minerals and water movement in the plant completely takes place with the help of osmosis. In our body, it is useful to remove the waste from the blood. With the help of kidneys, as you know, we are removing the wastage. That wastage is removed from the blood with the help of osmosis process only. Okay, then absorption of water in our body is also taking place by osmosis process. Okay, so this reverse osmosis process is studied by Friedrich Mercury and Bowie. These two scientists, Friedrich Mercury and David Bowie, and Friedrich Mercury, they studied the reverse osmosis process. Reverse osmosis process is nothing but it is a type of diffusion again. It is a type of diffusion again, but here we are using the plasma membrane. Okay, but we are using the plasma membrane here. So this reverse osmosis process is first of all used for desalination process. So what is desalination? Friedrich Mercury and David Bowie, they did this reverse osmosis process for desalination. So what is desalination means? Removing excess of salt from the water. Removing the excess of salt from water is called as desalination process. As you know, before the earth is covered by water and the majority part is covered by oceans. But we cannot use salt water. We cannot use salt water. We are using the fresh water. So excess amount of salt should be removed from the water. That process is called as desalination. The two scientists who worked on this is Mercury and Bowie. What they did is they prepared the plasma semi-permeable membrane, that is plasma membrane, in 3 layers. And they made the salt water to flush on this. So what happens here is, as you know, the definition of osmosis, movement of molecules from low concentration region to high concentration region. But here what is happening is high concentration to low concentration region, but not salt. Water is moving here, not the solute, but the solvent is moving here. Water is moving here. Hence it is called as reverse osmosis process. So what happened here? Salt water is moving on this plasma membrane, permeable membrane, semi-permeable membrane, which is made up of 3 layers artificially, and water is flushed on this. Which water? Salt water, saline water. So it is flushing on this. As a result, what happened? The salt is locked in this, and water will come out this side, and this water will become fresh water. This water will become fresh water and salt completely deposited here in the membrane. In such a way, we are extracting the salt, we are making the water fresh water from the salt water. Such a process was explained by Mercury and Bowie. They did this activity till now also. Salt water, that is sea water. From the sea water, the salt which we are eating, we are getting from sea water only. By which process? By desalination process. Till now we are following the same process called as reverse osmosis. Till now we are following the same process called as reverse osmosis, where desalination process is done, where this ocean water, salt water is becoming fresh water, and we are getting the salt for eating. By which process? By reverse osmosis process called as desalination, removing the excess amount of salt from the water. This salt we are using for eating purpose, for our purpose, for our domestic use, and the water is also becoming fresh here. Such a process is called as reverse osmosis process. Not only that, a very good example which we are using RO we are calling water plants. What is RO? That is nothing but reverse osmosis process. In every home, we are seeing this RO water plant, that is with water filters In olden days, we used to have the candles in the water filters, where we used to remove the impurities, excess amount of salt from the water, and we used to purify the water and we used to drink. And afterwards, we got the purine, where charcoal is there, it is removing the excess salt impurities from the water, and it is giving the pure water. Now we are using the ROs. What is the meaning of RO? RO is the reverse osmosis process, where excess amount of salt which are not useful to us is removed from the water and pure water is passed from the filters. So such a process we are using, the ROs which we are using at home, it is also nothing but it is following the reverse osmosis process to remove the excess amount of salt which are not useful to us and purifying the water and passing the water to us, the water which we are using for drinking. Okay, in our school also we have the water plant. What it is doing the reverse osmosis process and passing the fresh water for drinking purpose. Okay, otherwise it have excess amount of salt, where if we drink it is tasteless, means it is giving a salt taste, and not only that, it is if we take excess amount of salt, it is bad for our health. Okay, so in such a way, reverse osmosis is used in our daily life. Then last topic which we are going to discuss is dialysis process. Okay, dialysis process, it is there in 10th class in detail, but here also to study William Koch. William Koch was the first scientist who performed the dialysis process. Dia means through, lysis means breakdown. So, removing the impurities in the artificial form is called as dialysis process. So, who will undergo this dialysis process is the person who is having the ESRD, that is end stage renal disease, uremia. So, where both the kidneys fail, one kidney also functions, the person is alive with all the body functions and without any abnormality. But if two kidneys fail, such a condition is called as end stage renal disease. End stage renal disease. In such a condition where two kidneys become abnormal, they are not functioning, and as a result, the wastage is completely accumulated in the body, and the person undergoes the suffocation, excess blood pressure, heartbeat, all these abnormalities appear in the person's body. Then the person should undergo for dialysis process. It is nothing but artificial kidney process. It is also called as heme dialysis, hemodialysis, or heme dialysis. Means from the blood, hemo means blood. From the blood removing the impurities. From the blood removing the impurities. Such a process is called as hemodialysis. First person to perform this dialysis process is William Koch. Okay. Then what he did? A dialyzer he prepared. Dialyzer is a machine which is acting like the artificial kidney. Dialyzer is a machine which is acting like a artificial kidney. On that dialyzer, it is nothing but a machine. On that dialyzer, the tubes are connected, which are made up of cellophane. Cellophane tubes are there, this, and the blood is passed over this. Okay, so the person who is having both the kidneys fail, ESRD condition, that person is going this dialysis process where the blood is removed from his body with the help of artery. So from the artery, the blood is removed. Before removing the blood, the person is injected with the anticoagulant called as heparin. Heparin injection is given to the person. Then the blood of the person is removed from the arteries. As you know, which blood is present in arteries? Oxygenated blood. Oxygenated blood is present in the arteries. That oxygenated blood will become impure because of excess amount of creatinine, urea, uric acid. Okay, ions like potassium, chloride, all these ions, all these impurities are there in the blood of the arteries. Oxygenated blood also. So, with the heparin anticoagulant injection, the person blood is removed from the arteries and passed over the dialyzer, this machine. What this dialyzer is doing? This dialyzer is purifying the blood with the help of this complete setup of the tubes called as cellophane tubes. The blood is passed over this. This dialyzer is the tubes which are there, they are acting as semi-permeable membrane. These tubes are acting like semi-permeable membrane. These tubes are acting like semi-permeable membrane and removing the impurities and passing the blood in one bottle and in another bottle. The pure blood is collected here and the impurities are collected in another bottle. At the same time, simultaneously, this is connected to the person body by giving again the heparin injection here. The blood is passed into the person body by what? The help of vein. These are the two important points here. Blood is removed with the help of arteries and blood is injected or infused into the person body with the help of vein. Okay, in such a way, the dialysis process is done and for one sitting it will take 3 to 6 hours. Depending upon the severity, the person is undergoing this dialysis process once in a week, twice in a week, once in a month, okay, twice in a month in such a way. Depending upon the severity, the person will undergo dialysis. Why we are studying dialysis here is dialysis is also a process which is carried on osmosis. Actually, this process is done in a natural way in our kidneys to remove the excess impurities with the help of nephrons. Okay, structural and functional unit of kidneys are called as nephrons. Structural and functional unit of kidneys are called as nephrons. When these nephrons are not functioning, the person have to undergo dialysis process. So this dialysis, when both the kidneys fail, the person is undergoing the dialysis process. Okay, so this dialysis process will take 3 to 6 hours for one sitting, and the person blood is purified in an artificial way by the artificial kidney. It is called as dialyzer. It is a machine. Okay, so this process of dialysis follows the osmosis. Not only osmosis, it is also carrying on filtration. It is also carrying on filtration, removing the impurities from the blood by filtration process. This also I explained. Okay, so this is about our topic, today's topic. Now, if you have any doubt, you can ask.
This is the problem. One second, all of the more understanding is not there. It's like starting from chatting. Particular is completely wasted. When I am not directly related to class, we can interact. This is another problem. If trained with the law, you will not come to know. The person who is sitting here cannot be ready. Nobody will understand. You will say that everything is not clear. What I'm saying, we can understand. If you want to know that, you will getting the problem.